Good afternoon. Seeds contain within them the promise of new life. From the earliest days of genetics, the inner transformations of the seed, efforts to control genes and to alter heredity, have paralleled outward efforts in agriculture. This has remade our landscapes and our worlds and expanded our technosphere. As the Nobel Prize-winning American geneticist H.J. Muller announced in 1947, we cannot make life, far from it. Probably we can, however, remake it. We must not shrink from our destiny or fear it, he said. We must work in functional alliance with our genes so that evolution will become, for the first time, a conscious process. That will be, he wrote, the highest freedom that man or life can have, the highest form of freedom. Muller's mid-century hopes that evolution might finally be brought under human control closely mirrored the actual achievements of the so-called Green Revolution, that period from 1946 to 1966, when crop production more than doubled in Mexico, in China, in South Asia, and in other parts of the developing world, averting famines that are said to otherwise have ensued from rapid population growth. And indeed, the pure science of genetics and the applied science of agriculture have long been associated with each other. So seeds are fertile fields for thinking about life and its biotechnological possibilities. Today, treating and designing living things and thinking of them as technology platforms, our visions might include biofuels or pharmaceuticals. But over a century ago, efforts to understand and to control heredity developed hand in hand with the development of new fruits, flowers, and crops. The biological avant-garde has never been far from the avant-garden. The prospect of bringing evolution under human control was an enticing one for breeders and horticulturalists and geneticists alike at the turn of the 20th century. The dawn of a new mutation theory around 1900 brought these ideas of experimental evolution to the forefront in ways that might have commercial benefit, it was hoped. Researchers working in places like Cold Spring Harbor Laboratory in New York, which is still a major laboratory for molecular biology today, thought that they could hurry up evolution, that there would be a way that they could help the farmer to mold, they said, the animal and vegetable kingdoms to best suit his commercial interests, rather than to wait for nature to take its course. Creatures that a previous generation might have dismissed as monsters could now be carefully studied as mutants, in the hope that they would lead to, as one newspaper at the time put it, the modification and improvement of animals and plants at man's will. Although many had tried and failed to find a means of producing new mutants at will, the new discovery of radioactivity and of X-rays gave fresh hope. And it was in 1927 that Muller himself made a major discovery. In the course of a relatively simple experiment, zapping fruit flies with X-rays, he found that he had been able to increase the mutation rate and create hundreds of new mutants that had never been seen before. As many, he said, as had previously been found in the whole 20-year history of fly study, in which dozens of investigators had examined over 25 million flies. This was, he said, the promised land of mutations, where all types of mutations, large and small, ugly and beautiful, burst upon the gaze. They were a motley throng, he said. The roots of life, the genes, had indeed been struck and had yielded, he said. Muller's triumphal achievement, which he called the artificial transmutation of the gene in his landmark paper in the journal Science, spread far and wide and was said to have opened a new epoch of genetical research. And it was from mutant flies like these that geneticists began to understand the secrets of the gene, the relationship of genes to chromosomes, and the stuff of life itself, the seeds within seeds. Muller ultimately received the Nobel Prize for this work, in his path-breaking work in radiation, but many years later, in 1946. This is the year after the atomic bombs were dropped on Hiroshima and on Nagasaki, and therefore not coincidentally a time when many people were concerned about the effects of ionizing radiation on health, human or otherwise, and on heredity as well. Indeed, Muller's increasing concern over the years led him to consider mutation more of a threat to public health than the promise of a better future. Already envisioning the dangers of an increasingly radioactive Cold War world, in 1948, he called for mutational prophylaxis, a decrease in our overall exposure to radiation, whether from bombs or from medicine. But in 1948, a natural mutagenic process was also discovered, when Barbara McClintock succeeded in accomplishing with the genome itself what Muller had been using ionizing radiation to do. So McClintock had set up particular crosses that she had made in maize. Particular crosses where the chromosomes themselves would manage to tear each other apart, the things that radiation had previously done, and the chromosomes would then rearrange and rejoin in complex and interesting ways, but with no radiation required at all. Nature could be its own disruptor, its own designer. And it was in 1948, after 15 years of work on this, that she managed to confirm the existence of what she called mutable loci in maize, or what would later be called jumping genes, or also transposons. So this report that McClintock wrote in 1948, published in the annual report of the director of the Department of Genetics at Cold Spring Harbor, that same place I had mentioned, is where she had first produced this report for public consumption here, this mutable loci in maize. Why don't you take a look at it there? So a colleague who shared this with me said that reading her papers, which are incredibly dense, is a bit like reading mathematics, but upside down and backwards and in a foreign language. So very dense and complicated work that she managed to do. As an expert in cytogenetics, she had been used to looking through microscopes. This was her standard form of being able to learn things, right? To see what was going on with the chromosomes in the cells. But the effects of the changes that she was studying were sometimes also visible to the naked eye. Put some of these up here so you can take a look a little bit closer of some of the kinds of things that she was interested in. We're very lucky to have these samples here that I can show you that you can take a closer look at. And the ones that would not have made it across the border, I'll put on the screen up here for you to take a look at. So some of these kernels have sectors or dots or stripes and very interesting patterns that she learned how to read very carefully by telling exactly what was going on in each of these kernels. It was a remarkable skill that she developed that came from a very long time of studying these in great detail here. She found kernels that had sectors or stripes or dots that were split in half. On the image above, you can see some kernels that are exactly half, and that this was basically a way that she could trace through pigment the cell lineages in the kernels. She learned how to read them, these stripes, and she said it looked like this cell gained what the other cell had lost. I couldn't get it out of my head, she said. An extraordinary thing happened. It had gone wild. The genome had gone wild. So not only had McClintock found many more mutations than she had ever expected, but she discovered something even stranger through her studies, that it was particular genes that appeared to be causing this breakage, and that these streaks were then evidence of this transposition, and that these genes were dynamic, that they hopped from place to place in the chromosomes, from one to another, and from parents to progeny. People have said how pleased I must be, she said, that all this transposition is going on. And I said, don't take that for granted. The real point is control. The real secret of all of this is control. And she even called her jumping genes controllable elements. So McClintock believed that she had broken the gene regulation system, that she had thrown it all out of kilter, and so that all these spontaneous bursts of hundreds of mutations that she began to see were the result of mobile elements that were silencing the genes that they landed in or near. And so rather than having discovered jumping genes alone, which other people had also been noticing, she held that she had discovered a new system of gene regulation, a way of turning genes on and off, part of what today we might consider under the category of epigenetics. And she spent the next 30 years trying to figure out exactly how this system worked. So while Muller had begun by focusing on genes and discovered these untold rates of mutations in fruit flies, McClintock began by manipulating chromosomes and ended up discovering these untold genetic mechanisms of expression. A few years later, from 1957 to 1966, McClintock began to work with associates in Mexico. This is the ancestral home of maize, of course, and she was there on a project to trace the evolutionary origin of maize and the complex history of plant and human associations. And this work had paralleled a larger effort at the time to develop new seeds for local distribution to farmers. So once again, genetical and agricultural interests were never far apart. As part of a committee for the preservation of indigenous strains of maize, McClintock began to understand how these different chromosome combinations, she said, could produce the most extraordinary plants, and ones that don't look anything at all like maize. I began to realize, she said, that any genome can make practically any other. Everything is ready for it. There were all the same kinds of basic things, she said, and it's just how you put them together, like an erector set, or Legos might be the example many of us are familiar with. We are now recognizing, she said, the extraordinary integration of the genome. So what I find interesting here is that the possibilities for a future synthetic biology were grounded both in an understanding of these remarkable hereditary mechanisms of nature and also in her experience in farmers' fields in Mexico. Seeds are nothing without environments, of course, and so it's also important to consider McClintock's own institutional soil. Most of her work on maize had taken place at this laboratory, Cold Spring Harbor Laboratory, the same place where these other researchers had worked to accelerate evolution, and in later years where other researchers worked on inbreeding lines of maize in order to produce this high-yielding modern hybrid corn. And her work in Mexico, the Mexican context, was conducted under the auspices of the Rockefeller Foundation, which had the explicit aim of creating what they called synthetic varieties for local use. So unlike American hybrid corn, which required the constant repurchasing of new seeds from a commercial distributor, the newly created Mexican synthetic varieties, and that's what they were called, and they were first released in 1948, these could be propagated through open pollination, and farmers could save their seed for planting the next time around. So while commercial hybrids worked well in the rich soil and the economy of the Corn Belt of the United States, the story was reversed in places like Mexico and different solutions were looked for. So biological innovations have distinctive associated political economies. In many ways, it was also around 1948 that Promethean efforts to remake heredity for human ends encountered the world of the geopolitical in an even more striking fashion. Nowhere was this more true than in the Soviet Union, where genetics and geneticists suffered a rather distinctly different fate. While in the West it was the manipulation of the gene that had been a central feature of genetics for decades, manipulation of the environment, large and small, took on a special form in the Soviet Union under Stalin. On October 20th, 1948, Stalin put forth a decree known as the Great Plan for the Transformation of Nature. This sought to remake vast landscapes by planting an enormous shelter belt of forests, as it was called, covering nearly 6 million hectares. This was an effort to plant trees to block dry winds coming from Central Asia that were thought to be the cause of drought. This great plan has been called the world's largest ecological engineering project, and also the world's first state-directed effort to reverse the human-induced climate change that had been observed. It was certainly the first to have a symphony, Shostakovich's The Song of the Forests, which was written in 1949 to commemorate both it and the great gardener, Stalin. One of Stalin's favorite advisors on genetics and agriculture went so far as to advocate for the cluster planting of trees, with seeds planted closely together, as if in affirmation of the well-known truth that Soviet comrades support each other. Over 60 to 80% of all of these government belts and forests were then cluster or nest planted with these tree seedlings very close together. And 8 years later, the result was clear. Seedlings in industrial oak areas experienced a 94% death rate, costing at least 1 billion rubles. Overall, only 1/3 of seedlings that had been planted by any method survived after 8 years. So the great plan for the transformation of nature was big on promise, but failed to deliver. The effects of Soviet pseudoscience on forestry were bad enough, but they were just one part of a larger Soviet approach that identified the manipulation of the environment rather than the gene as the key to the future of agriculture. In genetics, this went by the name of Lysenkoism, named after Trofim Lysenko, the peasant-turned-scientist who served as Stalin's advisor on these matters, and according to one historian who had taken off like a rocket into the firmament of Stalin's empire. Lysenkoism was in part a demonstration of Soviet peasant common sense as opposed to elite knowledge. And the story goes that when the distinguished geneticist Nikolai Vavilov asked Lysenko, well, how would you chemically distinguish the difference between a lentil and a pea? Lysenko responded, well, put them on your tongue. So Lysenko steadily gained power through the 30s and the 40s, but it was only in 1948, at an infamous meeting of the All-Union Academy of Agricultural Sciences of the Soviet Union, that Lysenkoist efforts at the kind of instantaneous remaking of Heredity that he had in mind became official party dogma. Denying the reality of genes as bourgeois fictions, you will never see a gene, for there is no such thing, Lysenko said. He claimed that all heredity could be molded by paying attention to human control of the environment. A simple technique called vernalization, for instance, could supposedly improve harvest by chilling and pre-adapting seeds to a harsh wintry climate, something that would obviously be helpful in the Soviet Union. But Lysenko made many other claims besides that stretch our credulity, that a pine tree could spontaneously transform some of its branches into a fir tree, that wheat could somehow become rye, that harvests would become ever more bountiful, whatever it took to make the truth of Soviet science apparent to the world. Lysenko's attacks eviscerated Soviet genetics for a generation. They delayed modern methods of agriculture, and they ultimately contributed to the starvation of tens of millions of Soviet citizens. And what were once common and Promethean dreams, both inside and outside the Soviet Union, to envision new ways to remake heredity for human ends, began to shatter under different political realities. As the sciences of heredity ran headlong into these other dimensions of geopolitics, Muller, who had long been a contributor to Soviet fruit fly genetics, fled the USSR. In 1948, he resigned from the Soviet Academy of Sciences. But his friends and his colleagues, who newspapers had called fly lovers and man-haters, suffered more difficult fates for their hereditary heresies. In the wake of the 1948 meeting, some 3,000 scientists lost their jobs, others committed suicide, and Nikolai Vavilov and several other distinguished geneticists were executed or exiled to the gulags. Lysenkoism dominated Soviet biology into the mid-1970s. But such ideological scuffles over science also left their mark on the very concept of mutation itself. In the Soviet Union, where mutations were conceived of under manipulations of the environment, mutations then began to be conceived as directed, as purposeful, as potentially useful, and often as being visible at the level of the phenotype. You would see it in the organism in the field. In the non-Lysenkoist West, and in no small measure because of Muller's important work on these issues, mutations began to be viewed as fortuitous, as deleterious, and as key factors in a new genetic calculus of risk that was based on the careful study of laboratory organisms. Protecting our own human heredity from harm was rapidly becoming not merely a scientific question about genetics, but one that reflected larger concerns about humankind's place within an extensive technosphere. For how long would our increasingly radioactive Cold War world remain habitable, Muller wondered, and he calculated thresholds of mutational load for populations. So important frontiers in biological research, whether in the field or about the atmosphere, were never far from larger political contexts in 1948. That the Earth was humanity's one shared home was becoming increasingly evident in other ways as well. V-2 rockets sent up to the lower limits of space offered new views of Earth. The clearest and most expansive photos of the curvature of the Earth, as it was described, were published in a magazine article with the caption, "So Columbus was right." And this newfound ability to see the Earth's actual curvature brought still further ideas for the transformation of Earth on a planetary scale, and even for the exporting of the seeds of terrestrial biology to other worlds. The ultimate escapist fantasy, and yet also the ultimate realization of an engineering vision of a proliferating technosphere. Older ideas of panspermia, that seeds of life were deposited on Earth from outer space, gained new currency as well in 1948, as astronomers and biologists publicly debated the possibility of life elsewhere in the universe. Following the novelist Olaf Stapledon's 1948 lecture on interplanetary man, within a short time, visions of life in space were intertwined with talk of seeding space with humanity in a new form of directed panspermia. It was also in the spring of 1948 that the newly emerging field of what was called astrobotany in Kazakhstan sought to use spectroscopy to understand the potential nature of potential Martian vegetation by comparison of spectroscopic studies of plants that were living in high altitude or subarctic environments in Kazakhstan. So studying how these plants maintained their thermal states in extreme climatic environments meant that there might be practical links between fluorescence, frost resistance, and drought hardiness in plants. This is what one Soviet researcher wrote, thereby showing the practical value of astrobotany. Even the most otherworldly dimensions then of the technosphere, the detection and the study of possible plants in space, could have direct agricultural relevance. Situating the emergence of life within a cosmic context was also part and parcel of investigations into the origin of life on Earth around 1948. And the same philosophical principles that could explain the origin of life in other worlds surely could apply to the origin of life here on Earth, or at least that's what the biochemist and Lysenkoist supporter Alexander Oparin had claimed. In 1947, Oparin denounced Muller in a full-page letter to Pravda and called for his expulsion from the Soviet Academy. But these links between the earthly and the otherworldly were just as compelling for Muller as they were for Oparin. To understand the cosmic history of the gene for Muller was to understand its emergence on other planets in our galaxy. In many other worlds, he says, besides ours, living matter must coexist, and that in the star-bespattered abysses of the cosmos, there must surely dwell a myriad exotic forms. Our Earth, in turn, may be poor in life compared with what exists on some of the even more favored planets of stars and other worlds, of other places, he noted. During a celebratory lecture on changing genes in 1947, and which was attended by over 4,000 people, Muller brought the terrestrial and the extraterrestrial together. He indicated an interest not only in the possibilities for humanity, for remaking the Earth, or for adventuring on other planets, but also the biological possibilities of remolding life around us. These were wondrous potentialities. If we could find a way to put our wishes in harmony with biological possibilities, he noted, then the world of plants and animals should increasingly be ours to remold as we choose. And by 1958, Muller concluded, strange winds are now blowing in from the atom, the gene, and outer space. So understanding the emergence of the seeds of life elsewhere could help us better understand how to remake the seeds of life here on Earth. The outer possibilities involved in understanding life's cosmic extent were mirrored by these inner hereditary ones for Muller. And this last year at the Library of Congress, I found an inscription in a book that he had published of his collected scientific papers, and which he had gifted to a young Carl Sagan. Muller wrote in the inscription in this book, about halfway down there, "What a triumph it will be when we can coordinate and combine our outer and inner quests." And then he says, "May we someday meet in spirit on the tundras of Mars." Is genetics elsewhere than on Earth another possible meaning of epigenetics? Lecturing under the title of Man's Responsibility for His Genetic Heritage, just a month after the launch of the first Soviet satellite, 60 years ago in October, Muller cautioned of the need for international cooperation. The world cannot afford to allow to individual countries, he said, their separate genetic Sputniks. So whether dealing with the nature of mutation and its prospects for agricultural production, or astrobotanical investigations of life on Earth and in other worlds, the scientific study of seeds of life has always been part, of course, of larger social, national, and ideological contexts. Agriculture and genetics, heredity and heresy, Earth and outer space, science and politics, time and again, we see the entangled links between seeds and their soils, between genes and their organisms, organisms and their environments, our inner and our outer quests. Direct manipulation and indirect ideologies are constantly transforming seeds and replowing the technospheric fields in which they grow. Culture and politics are interwoven in productive and provocative and sometimes problematic ways with biological innovation. They provide the seeds of possibility for new scientific understandings and for new forms of life, as well as some of their uprooting and their destruction. And what are seeds, of course, if not instantiations of a promise of life as it could be? Thank you. It's interesting for me as a historian of science to always get to study science and scientists and to speak with them, but I'm very curious to hear what Arren's thoughts are of the history that he's just heard.
So that was fascinating, thank you. You know, the first thing that comes into my mind listening to your history is that how much ideology influences science. So we brought up, or we taught, that we need to look at science as something really objective, something that, you know, you come out without any, like tabula rasa, everything empty piece of paper, and you want to, you know, think about hypotheses, how to check them, how to disprove them or prove them, and so on. But here you come and say that actually consciousness or not consciousness in our mind or not in our mind, actually our ideology, our politics is actually playing such an important role. So do you think that today as well here in the West, in synthetic biology, in agriculture, we are still prisoner of such politics and ideology?
It's an interesting metaphor to call it prison.
Okay.
I don't know if prison is the metaphor that I would use, but I think, of course, we live in the world as beings with institutional contexts, with funding possibilities, with questions that are of interest to us, and sometimes with the possibility to find that funding by answering questions somebody else wants, right? So it's not a prison that one is in, but it does condition, of course, the kind of work that we end up doing. So there is no escape from being a human in the world, is what that means. Now, that's not to say that things are automatically going to turn Lysenkoist at any possible moment. That's, of course, not the outcome.
They are
that one
not
would
like
want.
this now? That's the question. Maybe we are under a Soviet kind of regime of thoughts and we are not aware of it, because I guess that they were not aware of it back then. So.
Well, and one can make the argument that that's not too far from the kind of obviousness of hybrid corn in an American national context, for instance, and how very differently that story would look in Mexico, where producing expensive corn that had to be bought for every generation for the farmers to plant would just not be something that would work in a different system. So I think what that shows is that national context sometimes makes a difference for what sorts of projects are pursued, and surely national context is not the only thing that makes that difference, right?
So maybe what we need to do is to put things on the table instead of under the table, meaning that to be aware that we are influenced both by politics, ideology, as well as our personal trajectory in life, and not try to be completely objective, not claim that we are objective, but actually work under the assumption that we are, you know, human beings, as you said, rather than, you know, machines that test hypotheses and so on. Maybe we need to change the way we actually teach children how to think about science.
Well, you would know this better than I do, but I have the impression that science is very hard already as it is.
That's true.
And so there's a great deal of training and work that goes into being able to accomplish the things according to the methods that need to be done. But there's certainly room for other ways of thinking about how science is done, and for other people who study science, not as scientists do, to point out the assumptions or the framings or the contexts that begin to produce the kinds of conditioning of the work that we see. And so it would be lovely if every scientist were as open-minded in that way and as well-trained as you seem to be, to think of all these other dimensions of science in addition to the very technical ones. But I don't think that's a requirement for scientists to need to do that. There should be enough room in our larger ecology of research that we can have places for many different kinds of work and that they can be mutually informing of each other.
Mm-hmm.
That would be my thought.
Let me move on to another point. So you mentioned also very interesting for me the thinking about life in outer space. Not just, it's something very, contemporary. In the last 10 years, people are talking all the time about finding the indication, exoplanet and so on. But it's very interesting to see that
and it is remarkable. Exoplanet discovery is something
indeed.
that has long been thought of, but to actually have the more than 4,000 confirmed instances of exoplanets is an exciting moment for that field.
And if you ask scientists from NASA, they say that, within 10, 20, 30 years, we will see the first evidence from life from outer space, whether it is in our solar system or indicators in other solar systems.
And that is interesting how those 10 years keep on receding as we go forward in time. This also happens.
Exactly like cold fusion. Yeah, it's always 20 years from now. That's true. But the question is, how do you, as historians or historians of science, see this evolving, the notion of how do we need to think about, life in outer space? Because this is the ultimate seed, the seed of all, the seed of life that starts everything. So how this evolved in the last century?
So that's very interesting. One of the things that makes me think of is today a lot of the language is that's being used is of habitability,
Yeah.
that we might find a place that is habitable. And what does that mean? Many times that seems to mean that it would have liquid water, that it's in a habitable zone surrounding a star. And that concept goes back to the 1950s, certainly, and back to the late 19th century, and it ties in with larger histories of colonization on Earth, the idea of habitable places for people to live. If we look over the century and we go earlier, we can see things like the Rand Corporation producing a report called Habitable Planets for Man. And so the idea of a place where life could live also being a place where humans could live, and that colonization idea was baked into that idea of habitability at the height of the Cold War is interesting. If we go earlier back in time, we have the appropriation of the language of humanoids from science fiction that's being used. And earlier than that, it was men of other planets and other worlds. So there's a story we could tell of going from these ideas of life in other places being that they would be like us, to they'd be kind of like us, to, well, maybe we'll find some animals there. A silicon giraffe or something is what Carl Sagan joked about on Mars.
So they see life there as a very similar or mimicking the lives here.
As very similar, except it gets smaller and smaller as you go on. Then it becomes, well, maybe we'll find just some plants or some algae. And then it becomes, well, not even life itself. We don't have to find life there to feel successful in the quest. We can find habitability. And I think that's an interesting story if a historian were looking at this to be able to
Well,
to show
I would
that.
claim from my perspective that thinking that we'll find something which is similar or mimicking what we have here is very, very naive. I could use even worse words than that because, I'm going to talk about it, about how chaotic is the process of life itself, how minute changes in the starting condition or the environment can lead to completely different results. So assuming that, another trajectory of evolution for 4 billion years will eventually lead to giraffes is almost ridiculous. Yeah.
And this has been an interesting debate in the field for a long time, where a paleontologist, G.G. Simpson, back in 1960 or so, wrote about the non-prevalence of humanoids in the universe. And what he knew from the history of paleontology was it was a history of contingency, that there was nothing to be expected that would ever happen again the same way twice.
Yeah.
But this idea that there could be commonalities of life here and seeds of life as they might have emerged in other places, either from a common origin or because of some other convergent feature of evolution, has been a part of actually practical efforts, and what got called, in the American context at least, planetary protection, that you'd want to make sure you don't bring back bugs from the moon and have them brought to Earth and cause some sort of pandemic here, And so that idea...
Now it's, of course, the opposite.
And now it's
don't want to win our pandemic to outer space.
That's right, or we don't want to contaminate some other place with seeds that have come from Earth that would then make it impossible to know what the indigenous life forms would have been in another place. And of course we can, and there are scholars who've looked at this in wonderful ways and who've said that language of barriers and of preventing contamination is echoing the language of containing communism at this time, that everything deserves its own protective
Which we go
sphere.
back to ideology again. It's exactly like, the Soviet notion that everything can transmute to everything. If I'm going back, it sounds to me very Soviet, Everything is equal, everything can, everything has the potential to become everything, while in the West, they focused about individuals, about individual species, individual genes, something which is, very West and not very Soviet. So
well, what I also find interesting about that story is that the dreams of what I call Prometheanism on both sides were shared dreams, these ideas that you'd be able to remake living things according to whatever your desires were, and it might not be that you'd produce wheat from rye immediately, but certainly you could produce a better form of wheat, a better form of corn. These ideas are shared across many different contexts in earlier years. What Lysenkoism does is it creates a fissure. It makes it impossible for those kinds of earlier Promethean sorts of claims to be taken seriously among geneticists in the West because it starts to sound too much like Lysenkoism. And you can see that in letters that they write, things that they start to discover other forms of systems of heredity, plasma genes, and things like this, that one has to be very careful how one describes heredity so that it doesn't end up sounding like something that's now become problematic in very complicated ways.
So from my perspective, in spite of the big difference between the West and East in this regards, they are both still trapped in the trap of tinkering, which I'm going also to talk about. Basically, taking something that already exists and manipulating it, interbreeding, changing the environment, but there is still no engineering there. No one of them, they could have dreamt about engineering, but none of the stuff that they do is really engineering. It's more like, it's playing, it's seeing what's happening if you press here, if you do that. It's like tinkering, textbook approach of tinkering. So how much these people 50 years ago, 70 years ago, really thought that we are going to stand here at this stage in which we can do actual engineering, not just tinkering.
So that's where the historian would say, well, a lot of people have thought that they've been doing engineering for a long time.
Okay.
You might not like to recognize their way of doing it as fitting what you understand to be engineering, but they called what they did engineering. And of course, they knew what engineering was at that time, right?
I have
and
a
so
problem
we can,
with that,
we can
yeah.
look, the phrase genetical engineering dates back to the early 1930s.
Okay.
The 1970s use of the term genetic engineering initially meant eugenics, and then became applied as the generic term for recombinant DNA work that was being done.
Yeah.
And now we have these new fields that I think we'll be hearing more about as synthetic biology, where these ideas of properly engineering things come back. But certainly we can trace the history of trying to engineer life right back into the late 19th century, and that was the term that they used.
Well,
However,
I would claim, one second, I would claim that if you don't know what you're doing, you're just playing around, but you don't know what you're going to get, and you don't have the mathematical framework to design it, you cannot really call it engineer. And they didn't have the mathematical framework, and they didn't know what we are going to get. They just, play around to see what's happening. So I think that, it's almost a dictionary level of what is engineering, and I think that if they call themselves engineers, they were really too optimistic about what they're doing. I think
don't know
that
if they called
even
themselves.
us are too optimistic these days about the level in which we can actually, take a system and completely engineer it.
So that understanding of engineering being defined in this particular way and that they didn't have it, I would prefer to see it like the story of When Will We Discover Life? That there's this shifting idea of what counts as proper engineering, and that changes just as our technologies have been changing as well. So I think that would be a way to both understand why somebody in the 1890s could say, well, to properly understand life would mean that one would be able to produce it from scratch in the test tube, and that is the ultimate goal someday of an engineering biology, that would carry right through the 1930s, 1970s, and to today. It doesn't need to be the same engineering in all those times, but I think that historical process of what counts as a properly engineered approach to biology is an interesting phenomenon, at least for the historian.
Thank you, Louis.
Yes, wonderful. I look forward to hearing.